Hi, my name is Ashley Volts with Antibody Engineering, and today I'm speaking with Ben Coverdale, Account Director at PatSnap.
Thank you for speaking with me today. Tell me a little bit about yourself before we get started.
Of course. Well, my name is Ben. I've been working at PatSnap for around 5 years now. So one of the account directors here in the life sciences team. And I have a PhD in biomedical materials. But now I'm working in the life science sector, like I say, helping companies to develop new therapeutics and explore the landscape around drugs and new developments.
Great, very interesting stuff. Let's get in to talking about PatSnap. How has PatSnap seen the antibody and biologics industry pain points change over the past 2 years?
Well, it's a very good question. And over the last few years, we've seen a massive change from the sort of wet lab experimental approach, where it's very laborious and time consuming and costs a lot of money, and to the advent of AI, where we're doing things very, very quickly. But even over the last couple of years, where we've been able to use AI, actually validating the data has been one of the main pain points that we've seen. It's all very well getting an LLM to read through an awful lot of this information that you're producing. But at the end of the day, you need to derive actual insights from this information, and understanding what is real and what insights you can trust is a really important thing to get a hold of, especially today.
Yeah, absolutely. And what are the biggest challenges antibody R&D teams face today, and what shifts do they need to make to stay ahead of the curve?
Yeah, very good question again. So it comes back to that, what I touched on before with the quality of the data. There's so much data now. It was a real problem before. Data scarcity was the issue in this field. And now there's a lot of data, but how do you validate it? What is the way that we can actually pull insights out of this information? And that occurs because you get things like siloed information stuck in different teams. And maybe you're focusing on one particular type of data, but you're not looking at how it links into other aspects of how that could be important. So I'd say connecting information together, understanding how all of these different insights can be linked across not just one division of a business, but multiple different areas, are ways we can extract the best insights and overcome these challenges.
Oh, that sounds great. Now, how about AI? Everyone's talking about AI. Where does PatSnap see how agentic AI can have the greatest impact for scientists and upstream R&D teams? And what are the common pitfalls?
Well, AI is a very useful tool, certainly used in the right context. And I think one thing that PatSnap is very good at is using AI to do things like extract key information. So one of the things we can do extremely well is pull data out of patents. And one of the things that's particularly relevant to the Antibody Engineering and Therapeutics Summit is being able to extract not only the antibody, but the corresponding antigen and things like binding data and relevant experimental information as well. Now, we do that very, very well. We've been working for years to train our algorithms to extract this information. And we validate this with our team of engineers who've been working on this for many, many years now. So that's where PatSnap does a very good job. What I would say the common pitfalls is that people tend to charge into this, where we hear this quite often. Bad data in results in bad data out. How do you possibly expect to make good, impactful decisions if you're not using the right data to do that with? So what PatSnap is really focusing on is developing very accurate and reliable information sources that development teams can reliably make informative decisions from.
Great. So PatSnap is highlighting the data foundry concept for biotechs that may lack these in-house capabilities. Can you shed some light on the importance of consistency and extraction challenges?
Well, yeah, if you think about how things have shifted nowadays from, like I said, the experimental methods, where it's a very slow process, but you have a validated method of doing things. You'll create a load of experimental data under the same conditions, and you'll use that to sort of benchmark your future experiments, your future results from. And we're just supercharged this with AI. We've enabled the ability to pull out more and more data. And now we have these volumes of information that we can make more impactful decisions from. Great. But in that information, how can we structure this and understand the data more? We're not so clear on whether this information has been derived from an experiment which was pulled using certain conditions. Like maybe they've changed slightly in a new patent publication, and they haven't really divulged what information that could be. So yes, we do have a lot of information to go at, but understanding the nuances within the data and how they change across different data sets can cause a lot of variability in the results. So whilst there are sources of information out there, so I mentioned antibody and antigen pairings before, it's important that you use a validated source which has been curated properly. And that's what a lot of our focus is to make sure that all of this is standardized, not just the data output, but things like the ontologies, the naming conventions, the experimental conditions, so that researchers and teams developing these new therapeutics can have the best insights and understand exactly what these new therapeutics are doing and what they're capable of when we move from a theoretical in silico environment into an experimental wet lab environment.
Interesting. Okay. And the recent buzzword is no longer just speed to clinic, but improving overall success rate. How should organizations think about this so all teams can move faster? And what is PatSnap's experience in these?
Well, absolutely. Before, again, if we go back just a couple of years, you want speed is everything. So this is where the AI race began, and we saw all of the new LLMs popping up from left, right, and center. And that's great. We're getting a lot of quick decisions. We're getting quick ability to develop these new therapeutics. But how many of them actually get through to the clinic? How many of them are actually validated candidates? The answer is not very many at all. So in order to improve the profitability of organizations and get therapeutic candidates into the clinic, we need to improve the success rate. And often that is a very, very low number, especially when we're developing theoretical antibodies which don't even exist yet. They might not even be possible to exist, but they're not quantified. We can't possibly hope to make new therapeutics out of them. So yes, absolutely. The challenge now is bringing in multiple data sets, not just the information we're finding in patents, but multi-sourced evidence where we can combine this information that we're collecting to make the best understanding and interpretation of which of these candidates are actually going to give us a good probability of success. And that's what we're seeing nowadays with a lot of organizations. A lot of our clients that we work with that is their number one objective. Yes, we've done the speed and we've set up the in silico teams, and we've got our huge data crunching computers to basically try and develop these new therapeutics. But now we need to understand which of these we're gonna select and take forwards, and which ones have the best potential to be therapeutic candidates. And that's the big challenge that we're all facing today.
Yeah, sounds like it. Anything else you want to add?
Anything else I'd add? I would say over the last 20 years or so, PatSnap have been at the forefront of not only patent analytics and sequence extraction, so for things like peptides, antibodies, and biologics, but we are the leader in life science AI intelligence and actually making this sort of data meaningful. So not only do we have that SaaS capability in the platform, but we have the data application as well to help teams who are developing these new therapeutics. So if there's anyone who's interested in learning more about PatSnap and what we can do, reach out. Very happy to chat about your ambitions and what you'd like to do and how we can support.
Great. Thank you so much for speaking with me today, Ben.
My pleasure. Thanks very much.